Hi, my name is Steven Giblock and I'm a senior at Maryville College. I'm doing research on the sand fiddler crab, Giblock Pugilator, here in Beaufort, South Carolina.
Giblock Pugilator is a species of fiddler crab found along the coastal estuarine salt marshes of North America and the Gulf of Mexico. I also came across Giblock minax, the red-jointed fiddler crab, and Giblock pugnax, the mud fiddler crab.
The purpose of my study is to find out if Giblock Pugilator can be used as a sentinel species for estuary contamination.
For my study, I found 3 different sites. First, I had a reference site which was unpolluted by human impact. Second, I had a site impacted by municipal pollution. And thirdly, I had a site that was impacted by a nearby golf course. The reference site exemplified a habitat that was virtually undisturbed by human activity. The Lemon Island Preserve is a large plot of protected estuarine salt marsh on the southern side of the Broad River. The municipal site was located on Parris Island, which is a portion of Port Royal. This site was an example of municipal impact as it received effluent from the Parris Island wastewater treatment plant, as well as the unlined Causeway landfill. The golf course site was located on Hilton Head Island just near Harbor Town. This site was located along the banks of the Hetty Gutter Creek near the Deer Island Road Bridge. This creek is the sink of the Harbor Town golf course. Crabs from each site were collected by hand and were marked with a Sharpie so that recaptures could be identified.
First I will look at the crab's morphology, comparing their carapace widths.
Each crab was carefully measured at the widest part of their carapace using vernier calipers.
I will also be comparing the male's dominant claw to their subordinate claw.
The dominant claws were carefully measured from the tip of the immovable finger to the very base of the claw. The subordinate claw was measured just the same, and then a ratio could be calculated of dominant claw to subordinate claw size.
Secondly, I will be looking at the population dynamics at each site.
A plastic ring was randomly thrown within the boundaries of the burrowing area of the site. Then each of the burrows were counted within the ring. This was repeated 90 times per site so as to get an accurate depiction of the population. Water samples were collected from the nearest body of water to the site, and great care was taken not to disturb the water while the collection took place. The crabs at each site were also observed for any signs of mating behavior, such as this, the claw wave. Around mating season, males will often wave their large claw in order to attract mates. The claw wave also functions to ward off other males that are competing for mates. The environmental data showed that salinity and recapture rates were found to not be significantly different between any of the sites. However, temperature was significantly lower at the golf course and turbidity was significantly higher at the municipal. Data was analyzed using a two-way ANOVA, which showed the significance of site, date, and the interaction of site and date. The carapace widths were shown to be significantly different at all 3 sites. Population density was also shown to be significantly different in all 3 sites. There was no significant difference in claw size ratio between the 3 sites, and there were no clear patterns in this data. The claw wave was rarely observed at both the reference and the municipal sites. However, at the golf course site, it was witnessed on every single day of observation. Practically every male crab at the golf course site was witnessed doing this claw wave. In conclusion, I found that the individual morphologies were significantly different at all three sites, with the crabs at the golf course having the smallest carapace width. One unexpected finding was that male crabs at the golf course site exhibited increased claw wave behavior. For population comparisons, both of the impacted sites had increased population densities. We cannot implicate any particular anthropogenic impact as causing these differences, as no water analyses were done. However, this study is the foundation of future studies investigating Giblock Pugilator as a bioindicator of anthropogenic impact.